Hello, everyone. Sorry, there was a technical difficulty. We're back with our 24th Journal Club with Antibodies. And joining us today is Dr. Mark Anderson, who will be here soon, and Dr. Jeremy Warschauer, who is here right now with us. Welcome to the Journal Club, Jeremy.
Thank you. Thanks for having me. I'm Jeremy. I'm an endocrinologist along with Mark over at UCSF, and we're excited to present our immunology-related research focused on autoimmune disease, and in particular type 1 diabetes today.
Thanks a lot. And without any delays, I'll hand over the virtual mic to you so you can present us the work.
Great. So here's our article we'll be presenting. It was recently published in the Journal of Experimental Medicine in their August issue. We're really excited and happy about that. A lot of great people really helped contribute, and want to just call them out here. At the beginning, didn't want to save it for the acknowledgements at the end. But yeah. And just to get started, I wanted to give some background on type 1 diabetes, which is caused by a break leads to a destruction of pancreatic beta cells. And it results in absolute insulin deficiency and high glucose levels. The two hallmarks of this autoimmune disease include the presence of islet autoantibodies that are highly predictive of disease development, as demonstrated in this figure here on the left. And the number of autoantibodies you have seems to also predict kind of your probability of developing diabetes over time. As well as insulitis, which is seen here on the right, with the red arrow indicating the lymphocytic infiltrate into the islet, which contains the insulin-producing beta cells. And so these get obliterated and you develop type 1 diabetes.
Mark, when you say number of islet antibodies, does that mean how many islets are positive for antibodies?
No, so in this test, you're basically taking people's blood and you're looking for serum autoantibodies. And there are 4 that are classically tested in clinical setting. And so some people maybe just be positive for 1, such as GAD65. And you can see here, they have a relatively low risk, but still it's greater than no autoantibodies being present for developing type 1 diabetes. But other people may have GAD65 and insulin autoantibodies, and those people are at much higher risk for developing type 1 diabetes over time. But it's serum autoantibodies.
Okay.
Because we don't have any great way to non-invasively look at each islet individually.
Okay, thanks.
Mm-hmm. And so to kind of understand this disease and what genes are important and understand mechanisms, there have been 2 approaches. The first approach are these genome-wide association studies that we often talk about. And they've been able to show that type 1 diabetes is a polygenic disease with the majority of its genetic risk attributable to this HLA allele in the MHC region. And so for those less familiar with the HLA, this is part of the antigen presenting cell that displays peptides to the T cell and forms part of what's called the trimolecular complex. So the T cell, if it's a good match, it gets activated and it kind of do its thing. But there are numerous other susceptibility alleles like insulin, CTLA4, PTPN22, just to name a few that have been identified from these GWAS studies and help estimate individual risk. But what's important is that these are kind of associations, but we actually need functional studies to determine what is their significance in terms of diabetes specifically, and are there direct pathways that actually help map out. I think a lot of those studies are kind of TBD at this point. And so we're really interested in understanding what are direct mechanisms driving the disease. And we think that naturally occurring cases of monogenic type 1 diabetes, although really rare, things like loss of function mutations in AIR or loss of function mutations in a gene like FOXP3 have actually been the most informative for understanding how central, as in the case of AIR, or peripheral, as in the case of FOXP3, tolerance mechanisms break down. And I think they also have been kind of the basis for a lot of therapeutics that have come through development and exciting opportunities. So we really like what the post-GWAS studies, which are kind of these broad population-based ways of finding hits of interest. These we really think help deliver very targeted insights about type 1 diabetes. There's something important which you may be thinking, that's type 1 diabetes hasn't really been cured, and there's been a lot of work in it. So it leads to the natural question of, are there other monogenic forms of this disease that may really help further shape and refine our understanding of type 1 diabetes? And so with that kind of leads to this patient, which really fit this profile of somebody who might provide deep insights into this disease. And so the patient basically had neonatal type 1 diabetes, a very extreme form of type 1 diabetes, in that they were diagnosed at about 2 weeks of age. And in addition, they presented with celiac disease, autoimmune hypothyroidism, early onset, autoimmune disease, multi-organ. And so leveraging the power of whole exome sequencing, researchers did a simple, elegant, and extremely informative experiment. They basically sequenced the patient along with both of their parents, and that allowed them to kind of see the difference in the genes between the two. And there are all these other filtering techniques you do when you're performing whole exome sequencing, because there are lots of little changes and variants that really don't have any significance. And so you have to know how to filter those down really effectively and sift through all of the noise. But when they did all that, they came to a variant in the gene STAT3, and kind of a new finding. And they weren't exactly sure, I think, if that was truly the causal variant, because it would have been unexpected. So they did another round of these experiments, and basically took a database of individuals and that presented with similar kind of outlier autoimmune diseases, but these kind of severe outlier diseases. And they just sequenced the parents and the individuals. And there were 4 other individuals that popped up with mutations in STAT3. And this was really exciting, because it may mean that this is driving the autoimmune diseases. But what was challenging at the time was, before this, all of our knowledge of what STAT3 mutations led to was in the context of their loss of function. And this results in something called Joe syndrome, which is, you see these hyper IgE levels and a lot of these kind of features shown here on the right. And this was in total contrast to what these patients were presenting with, which was an autoimmune phenotype. And so that's the question of what's going on. So to figure this out, they basically did a luciferase assay and found that these weren't loss of function, but instead they're actually hyperactivating mutations in STAT3. And one in particular really caught our attention, and that was this mutation K392R, because of its really extreme activity, and the fact that this patient was essentially diagnosed with type 1 diabetes at birth. This is really just such an extreme form. We thought that studying this could really lead to some great insights. And so for those a little less familiar with what STAT3 is and what it does, it serves as a transcription factor, really famous for being one of about 5 stats that make up the famous JAK-STAT signaling cascade. So within the immune system, usually a cytokine like IL-6 binds its receptor, the JAK protein phosphorylate, which results in recruitment and phosphorylation of STAT3, dimerization and translocation to the nucleus, or combined DNA and active transcription factor for genes like IL-17 or SOX3, just to name a few. And so we wanted to figure out how excessive STAT3 signaling leads to type 1 diabetes, but it's important to call out a key challenge is the fact that it's ubiquitously expressed in the body, in cells. And that's including CD4 cells and CD8 cells, but also MTECs, which are in the thymus, key cells involved in central tolerance. They express STAT3. The pancreas, it expresses STAT3, many of the cells. And so it's really not a trivial problem to understand what it's doing because of this ubiquitous nature of its expression. So we really knew we had our work cut out for us, but felt if it could lead to the type of insights other Mendelian forms of autoimmunity have provided us, like FOXP3 and AIR, then it would certainly be worth the effort. And so it was a bit of a risk, but we thought there's a big reward in pursuing this. And so the fundamental question we wanted to answer was what's the mechanism driving STAT3 gain of function in type 1 diabetes development? And so to approach this, we decided to use CRISPR to create a STAT3 knock-in mouse in the non-obese diabetic mouse model. And this is the classical type 1 diabetes mouse model that we use in research. And we leverage it because there are lots of similarities to human type 1 diabetes, and as well as many genetic tools that we can leverage to enable us to give kind of deeper insights much more rapidly using the model system. And so on the right, you can see confirmation by Sanger sequencing that our mutation was successfully incorporated germline into the mouse's DNA, and it was in a heterozygous fashion. So just like the patient, this was very identical. And here, you can see the mouse on the left. By the way, can y'all see the zoom bar that's moving around?
You mean the cursor?
Yeah. Or there's like, well, I think it's okay. Y'all haven't said anything. So here you can see our mouse on the left. It's a little smaller than the wild type. And this is actually really interesting because in almost all of these patients, short stature is noted and kind of growth retardation in the humans. So this fact that they're smaller was a really good sign that this mutation might help recapitulate the human phenotype as well. But we wanted to confirm the STAT3 activating behavior of this mutation was conserved in mice, especially in the immune compartment. And so we took naive CD4 T cells. We isolated them from lymphoid organs like the spleen and other lymph nodes. And we differentiated them in vitro into Th17 and Treg subsets. And the STAT3 T cells exhibited this increased differentiation into the Th17 cells relative to the wild type, while differentiation into the Tregs was reduced. And these were exactly what we would expect given the gain of function behavior based on what's been noted in humans previously. And so this is a good sign that, again, our model, at least in the Petri dish, was behaving as anticipated. So the real question is what happens in vivo? And so we kind of just went for the gold and said, do these mice get type 1 diabetes just like the humans do? And we basically just did weekly blood glucose checks on these mice, a lot of mice, I will say, and found both in the female mice as well as the male mice. Sure enough, diabetes was accelerated when you add in this mutation. And so this was, I think, one of our most exciting findings, the fact that we now had a mouse model with the exact mutation that's seen in humans, and that also was behaving in terms of phenotype, the same way that humans do. So we could really, our expectation is the findings that we were discovering and would discover with this model would actually be explaining what was going on in patients with this mutation. So one challenge is, given this really ubiquitous nature of STAT3 expression, including pancreatic beta cells, we wanted to just see, was there something going on in that compartment, that non-immune compartment first? Or was this truly an autoimmune thing going, driving the diabetes? And so we took young non-diabetic mice and we just did basic glucose tolerance testing on them and found it was totally identical between the STAT3 and wild type mice, suggesting the beta cell function was not being altered at all. We also took the pancreas, we did immunofluorescence. Here you can see this is an islet that we did immunofluorescence on. The green is the beta cells, the red are the glucagon producing alpha cells. And the appearance is really perfectly in line with how normal islets develop. There had been a thought maybe STAT3 induced this skewing towards more alpha cells instead of insulin producing cells, but we didn't find that to be the case at all. And so our findings suggested STAT3 wasn't interfering with the beta cell development or function, at least in our model system. Rather, diabetes seemed to really coincide with the presence of encephalitis. And you can see that here most clearly on the left, where we basically took mice at different ages and we took their pancreas, we did histology on it, H&E, and I would just count all different islets and see how many of them had lymphocytic infiltrates and score them. And we did this for a lot of islets, I will say hundreds of islets per mice. And this is a summary of the findings. But what you see is early on, when they don't have any diabetes, they also don't have any encephalitis. As you progress through time, the females do seem to get some encephalitis before they get diabetes. The males, it seems kind of like there's a switch that gets flipped, where all of a sudden it goes from no encephalitis to just really severe encephalitis at the time of diabetes development. And this made us confident we're dealing with the classical kind of encephalitis-driven autoimmune form of type 1 diabetes. And we also did mass cytometry of the islets, where we developed a kind of custom cytoph panel for staining islets, or not staining, but doing mass cytometry with the islets. And it allows us to do about 40 different markers that we thought would be relevant to kind of characterizing what was going on. And this is just the high level view, which you can see in the wild type mice. During the time of encephalitis, you have some alpha beta cells remaining, but you kind of see the early signs of B and T cell infiltration into there. In the STAT3 mice, when they become diabetic, you have none of these cells remaining anymore. They've all been obliterated. And you see this just huge influx of B and T cells into the islet.
Hey, Jeremy, I think Vijay's got a question.
Oh, okay.
Yeah.
Sorry about that.
No, I was just curious about the presence of B cells and T cells in wild type islet, because is it a specific, is it very specific to the STAT3 wild types, or
yeah,
because in at least in one of the papers I remember that it is mostly the residential macrophages which are in the islets, and or antigen presenting cells rather than B cells and T cells during the non-diabetic phase.
Yeah, you're totally right. Like very, very early on, it is a lot of those kind of macrophage dendritic cells that are hanging out there. You don't see many B T cells. I wish I had. I have a whole series of these STAT3 mice that we did this cytoph on, and you see exactly what you're saying. When you see all these alpha beta cells, you hardly see any B or T cells inside the islet. It's really just the dendritic cell kind of populations. And then as these start to go away, suddenly the B and T cells start to appear, and you get more and more until you basically get a figure like this for no more alpha beta cells, but really just lots of B and T cells. It really is an adaptive immune response. And within that compartment, the classical thinking is it's the T cell driven autoimmune disease, meaning T cells are typically sufficient to drive development of diabetes in this model. Yeah.
Which means
Mark,
that even in the wild type models, they do have B cells and T cells, is what this, like, at least from the T-SNE, that's what I understand. I don't know if I'm right or wrong.
Yes.
Yeah,
We
that-
didn't. Yeah.
At 8 weeks. At 8 weeks.
Okay.
Right. If you went earlier, you'd see fewer of those B cells and T cells.
Okay.
Are
Thanks.
these usual in non-diabetic mice? Are these exhausted T cells usually?
I'm
You're getting to
laughing
the
now.
punchline too quickly.
I'm laughing
yeah.
now.
Well, yeah, I think as we go along, that story will become more clear. Yeah. Maybe we
Okay.
can.
I had one
Alright.
last question, Jeremy. Sorry if I missed it. What is
Mhm.
the genetic background of these mice?
It's the NOD mouse model, the
Okay.
non-obese diabetic mouse.
Okay.
Yeah, that's important too,
Mm-hmm.
right? If we put the same mutation in a different strain, it wouldn't get diabetes at all.
Okay.
Yeah. Cool. So here, basically, we just started with some basic immune phenotyping using flow cytometry. And so to understand the cellular compartment, we kind of took the cells from the secondary lymphoid organs, the spleen, and did ex vivo flow cytometry. And what stood out to us the most was these rises that we saw in the more effector-like cells. Although I will say, even in the Treg compartment, we saw an increase in cells. And these are kind of consistent with our understanding that STAT3 is very critical in cellular activation and proliferation. So you would expect an activating mutation to be expanding. Although the Treg is a little different because based on the literature at the time, a lot of people were saying, oh, the hypothesis is STAT3 leads to a decrease in T cells. And we saw that in vitro. But as we looked in vivo, we actually saw something slightly different. Which just, I think, highlights what we were really excited that this model could deliver is in vivo findings come more closely translate to a human.
Yeah, well, this is an important point. This is an important point to like have just a little bit of a discussion about is, when in vivo, where we can actually look at like snapshots in time of what's going on in a highly controlled mouse model, right? These Tregs, we advertise them as the policemen of the immune system that helps suppress things, right? And what's really interesting about this NOD mouse model is when these Tregs first got discovered by my colleague at UCSF, in the NOD mouse, by my colleague at UCSF, Jeff Bluestone, that they were important. People did the simple hypothesis. Oh, I bet that the NOD mice don't have any Tregs. That's why they get diabetes, right? And sure enough, the first papers in the early 2000s came out saying exactly that. Turned out to be a big pile of crap, right? And then, right? And, it was one of these things like, I know what's going on, and I'm going to use these tools that aren't particular. At that time, the tools to measure the cells weren't very good. And, you were basically massaging the data to fit what you already thought, right? And then as time as time went on, we got better at measuring these cells. And I think the recurrent theme that we see is no matter what is wrong with the mouse, right? Unless they can't make Tregs at all, when they're about to get, when they're about to blow up with an autoimmune problem, they have tons of Tregs. They have more, right? And what it is, the lesson is that when you start perturbing with immune tolerance, right, these mechanisms start getting thrown into place to try to compensate and regulate it. right? So
Yeah.
going back to the data or Jeremy stuff, this didn't surprise me, but it does get against this little simplistic model that got built in humans when this disease got discovered, because it was something you can measure from the blood of the patient. But this doesn't say that the Tregs aren't defective. This is something. This kind of thing will always happen when autoimmunity is going on.
Just while we are on this topic of STAT3 and Tregs, there is an epigenetic mechanism through which STAT3 it reduces the FOXP3 expression, and what it does is it may not completely get rid of Tregs.
I don't agree.
You don't agree with that?
No. Nope. No, I don't know what's going on. Honestly, I think Jeremy's going to get into it here. The Tregs in this mouse work just fine, but of course, this is overexpressing STAT3, not underexpressing STAT3. Right.
Right.
Yeah, and it gets back to like what you just said, DJ, is the those kinds of experiments or things where they took cells that were naive, and then they gave, when you induce them to be a Treg, where you're giving cytokines, one of the cytokines they'll give is IL-6, which signals through STAT3, right? And so when you do that, and you monkey with STAT3, of course, the Tregs aren't going to work as well, right? But that's totally artificial, right?
Yeah.
Right. What's that kind of interesting too, since we're talking about this, is remember Jeremy had that slide of the yin and yang of this disease, right? There's this opposite disease where there's patients that are missing one copy of STAT3.
Yeah.
Right. Let's go back to that for a second. Yeah. The thing on the right. So this is called Job syndrome. Right. Are they blowing up with autoimmunity? Absolutely. Absolutely not. What their phenotype is, they have immunodeficiency, and they do have dysregulation too, but they get these recurrent bacterial infections. And then it's not even on this slide, but they get Candida, and it's been shown that they actually have a problem making Th17 cells correctly.
Yeah.
So anyway, that's why I'm sorry about this. I'll be a little dismissive about what you said, but I don't see it in vivo.
No, I was thinking maybe if you were to subgate on these Tregs with a FOXP3 high and a FOXP3 low population, you might see there's a lot of FOXP3 low population, and they're all just coming together as FOXP3 positive in these cells. I was wondering if you might. Yeah.
Yeah. I mean, we did that type of histogram within the Tregs. We didn't see anything. Maybe it was like ultra subtle, but
okay.
Yeah.
We didn't see that.
Yeah, that's another phenomenon. That's actually another phenomenon in immunology that's kind of interesting. As this whole subsetting of different T cell populations, right? You can have Th17, Th1. It's on the slide here, right? And a cool story that evolved in the 90s and early 2000s was they started figuring out what all of these transcription factors that make this stuff go, right? So Th1 is Tbet, Th17 is ROR gamma T, Tregs are FOXP3. And the general rule of thumb is once the cell commits to the lineage, right, all of this transcriptional programming happens to help lock it in, right? And so that's how you get these distinct effector populations of cells. And so I actually think this is complete nonsense when you start talking about FOXP3 high versus FOXP3 kind of high cells. That's not the way the system works, right? You actually get sort of this feedback loop where the cells become this nice tight population. And in general, you don't see evidence of like a cell that's a stable, weak FOXP3 expressor. Now in humans, when you activate a T cell, like just naive T cells, when they get activated, they'll turn FOXP3 on a little bit and then it'll go off again. And then some, depending on the situation, some cells will commit to the lineage. But again, you get sort of this stabilization of the amount of FOXP3. And so these kinds of, part of the reason I'm hounding on this is these kinds of concepts get legs because people write this or they say it with very little data behind it. Right? Yeah, you put cells in the fax machine and you can like make a cut wherever you want. But I don't think there's much data to say that there's really this business of like, oh, those are the low FOXP3 cells.
Cool.
Okay, keep going, Jeremy. Sorry.
Okay. No. These are great discussions. So one of the early experiments that we did was we made bone marrow chimeras, and so here we basically took a lot of these young immune cells from the bone marrow, from the STAT3 mice, and we irradiate some wild type mice, and then we transfer them in, do a bone marrow transplant, and then we see what happens. Do they develop type 1 diabetes any faster? And the idea being, all we've changed in this experiment is the hematopoietic compartment, but otherwise, the islets, everything else in the mouse is from the wild type, and so it really allows us to isolate whether this is just from that immune system compartment. We found it actually was sufficient to transfer diabetes. If we just transfer that bone marrow, that could drive the phenotype as well. We did another set of experiments, a lot of genetic crosses, where instead of asking, is it simply sufficient, could we, if we knocked out that adaptive immune system, for instance, by crossing STAT3 to the RAG1 knockout, essentially removing all the TMB cells from the mouse, would it develop any diabetes? And it did not. We did another experiment where we basically only changed the MHC from the NOD's MHC to basically the MHC of like the B6 mouse. And when you do that, all of a sudden diabetes completely vanishes. And so we realized this was an MHC dependent disease, and as well as kind of dependent on those TMB cells. But because of the MHC dependence, it seemed like the T cell compartment was probably very much necessary to see the diabetes that we were observing as well. And so I guess a question though that we hadn't answered yet was, okay, MHC expression is important, but is this something that's kind of intrinsic to the T cell, or is this more on the tissue expression side, that antigen presenting side of things, where the defect could be occurring? And so to figure this out,
hey Jeremy, can I stop? Can you flip back for a sec? Just so everybody's on the same page. This is like a great example, like how powerful the mouse system is, right? You can go in and just do this laser, this laser dissection. And one of the things we were dealing with, Jeremy touched on it a little bit, but one of the things we were dealing with this paper was, before we had published, another group had published a paper that they could take an iPS cell from this patient and then get it to be differentiated in vitro and get it to be a beta cell. And then they said, oh, this disease is not immune, it's a beta cell problem, right? So here, by putting the RAG knockout, that basically gets rid of all T cells and B cells. There's no adaptive immunity in that mouse, right? Mice don't get diabetes.
Yeah.
Right. So whatever these guys are talking about is probably incorrect. But what they'll come back to us and say is, oh, well, mice are different than people. Right. And yeah, they are. But when it comes to fundamental wiring, not so much. So I don't think there's an intrinsic problem with the beta cells here that's a major defect that's triggering diabetes. Yeah, VJ.
Like I was having this question, like what if in the same model, the macrophages or the dendritic cells were completely knocked out? Like, and
it's interesting. It's hard to do that.
Yeah,
it's really hard to do that. So people have done sort of experiments where you knock out some of them. Like an example of this is BATF3, turns out to be a transcription factor for a subset of dendritic cells.
Mm-hmm.
And the BATF3 knockout in the NOD mouse doesn't get diabetes at all. Mm-hmm. Right? Which just says, it says what they're important too, right? You need the antigen presenting cells to pick up the stuff from the islets to present it to the T cells and B cells. Yeah. That's right. That's right. Yeah. Another fundamental thing, this is another fundamental rule about autoimmunity, is almost any autoimmune disease that you look at in humans, and you do the genetics of it, the number one genetic risk factor that comes up is the MHC. It maps to the MHC, right? The HLA complex. And what it's telling you is that you have to have certain kinds of MHC types to get autoimmunity. It doesn't mean that it's going to define that you're going to get autoimmunity, but it puts you at risk, right? And so this H2B nomenclature, what that means is we put in a different MHC than the NOD mouse has, a different type, and now you get complete protection against diabetes, right? And so that just gets at like how the T cells actually see things. And so when you change the MHC to something else, the T cells don't attack the islets anymore, which is another fundamental rule about autoimmunity.
And this is a pretty drastic difference. No incidence versus.
Yeah, exactly.
Yeah.
Exactly.
And we did it like in Solitis scoring, and basically we saw no infiltrates. So
yeah. Well, you know,
the immune system doesn't get activated.
And this point, you know, look, this point I'm telling you, this is that, you know, you may feel like I'm kind of lecturing and this is like immunology 101. One of the reviewers of our paper said, oh, you need to put this mutation in a different strain of mice and get them to get diabetes. You know, like, that's ridiculous. They're not going to get diabetes. And it still doesn't mean it's not important.
Yeah. Yeah, I totally agree.
So don't discount what I'm saying right now. There's like even sophisticated professors get lost in this stuff.
Yeah. Our response was, well, we already showed if you only change the MHC to the B6 MHC, you already get rid of diabetes. So of course, if you put the whole B6 background, you also not get diabetes. That would just be overkill.
Yep.
We've already shown. Yep. Okay. Cool. So this MHC dependence, I think, raised an important question of, was it more on the T cell side? Like, was that more the driver, or was it more on that APC antigen presentation of the tissue that was kind of accelerating things? So we kind of did this by let's look at how the tissue affects T cells. And so we took T cells from NOD mice that had a transgene for BDC 2.5, which is basically an islet specific T cell. And we took those out from wild type mice. We labeled them with cell trace violet, which allows us to track their proliferation. And we adoptively transferred them into either wild type hosts or STAT3 gain of function hosts. So basically, these T cells would then be seeing the environment of that STAT3 mouse. And if it was the environment that was driving the T cell activation, then these mice should have a different proliferation profile than the wild type. They should be more highly proliferating. And we really didn't see that to be the case. In fact, proliferation was almost identical. It didn't matter which host we transferred the cells into. And so it made us think it's not really it's not the APC compartment that's driving this thing. We really think it's more intrinsic to the T cells. And we'll get to more specific experiments on that later, but this kind of, again, at this point, we're just trying to funnel down what is the appropriate compartment where we think disease is being driven. We didn't think it was that APC compartment. So we wanted to kind of figure out what was going on. And so we did what is pretty common these days, which is single cell RNA TCR sequencing. So we took wild type and STAT3 mice at about 8 to 10 weeks of age. And we came to the 8 to 10 weeks based on those encephalitis curves we showed, where that seemed to be that sweet spot of the mice weren't yet diabetic, but they had a little evidence of insulitis going on and that immune infiltration starting to take place. So we thought that would give us the greatest insight. Because once you get diabetes, it's kind of like cats out of the bag. It's almost impossible to really know what you're finding, if it's related to the glucose, if it's related to something else. So we needed that non-diabetic state. We took the islets out, and then we sorted just those CD45 positive immune cells and sent them off for sequencing. And I guess to provide deeper characterization, well, actually, just to start, on the left is the results of just the T cells that we sequenced. And what stood out to us when we compared the STAT3 versus the wild type numbers in the different clusters was this effector CD8 compartment, consistent with our early effects immunophenotyping, was really the compartment that seemed to be expanding most significantly. And so we decided to take these CD8 cells where the difference seemed the greatest and recluster them to try to dissect if there was something within those clusters that was really going on. And it ended up leading to these two very distinct clusters at a high level, the naive cells over here on the left, and then these more effector-like cells or non-naive cells over here on the right. And when we kind of looked at ratios of how the cells were distributed in those two clusters, it seemed like the effector cells, in terms of a relative frequency, were pretty similar, but it was more on this in the exhausted kind of compartment of the CD8 cells, where there seemed to be an interesting difference. That is, these STAT3 cells seem to get a little more biased to that transitory state, where they weren't pushing into the terminally exhausted state. They're kind of moving from effector to transitory, but then not really moving all the way to being terminally exhausted. And we thought maybe these transitory cells, maybe getting stuck in that state, along with the hyperproliferation activation, maybe that's what's driving this thing. And you can see the gene expression in the effector and transitory cells here. Really a lot of just kind of effector-like cytokines, CCL5, granzymes. Those were the ones that really stood out to us as being very highly expressed in these clusters. And we did FACS to kind of confirm. And yeah, sure enough, when we took like pancreatic lymph nodes and we checked to see if there's higher CCL5 or granzyme expression, that was the case. So the question though is, could these CD8s at the end of the day drive disease? Were they really, fundamental to the type 1 diabetes? And so we took the CD8 TCR sequencing part of the single cell experiment, and we analyzed that repertoire and found the 20 most abundant CD8 clones and kind of looked at them closely. And one in particular was very fascinating to us. And it was this one, 6158, listed here. This specific clone contains the CDR3 sequences that were nearly identical to those in the TCR 8.3 IGRP reactive TCR. That was known, and there's actually an NOD model with that already incorporated. And that's basically a CD8 restricted TCR specific for islet-specific glucose 6 phosphatase related protein. That's what it stands for. But it's known to be a kind of a diabetogenic TCR. The only difference between ours and that TCR is this one little serine, but otherwise the sequences were identical. So that was great. And it allowed us basically to, again, leverage this NOD model and all the kind of submodels that have been created over time, and do another genetic cross and test, are these CD8 T cells sufficient to accelerate type 1 diabetes? So we took our STAT3 gain of function mice, we crossed them over to the NOD mice that carried this TCR transgene. We got, the regular NOD mice with that TCR, as well as STAT3 mice with that TCR, and we just let them check them for diabetes and looked at their diabetes incidence, and sure enough, they got much more rapid diabetes than the wild type kind of litter mates, showing that it seemed like this was probably a driver, because this difference was a little more profound than we had seen just the non-transgenic mice, the STAT3 mutation. But we wanted to go even deeper, and because right now there's still lots of other T cells that are swarming around in these mice. They're predominantly 8.3, but they're not only 8.3, and there are all sorts of other cell populations. So we really isolated this system closely by taking only the naive CD8 cells that were primarily the 8.3 TCR transgenic CD8 T cells. We took them from wild type mice, and we took them from the STAT3 mice, and we mixed them with polyclonal CD4 T cells, which are necessary to induce diabetes. It turns out you can't just transfer diabetes with naive CD8 T cells. You need commonly called kind of CD4 help to kind of get things going. And this is an established way of doing the adoptive transfer as well. So we did this mixing of cells. We transferred them into NOD SCID mice, which wouldn't have any other immune cells present. And we looked at how their diabetes incidence curves shook out, and you can see just a really profound difference was seen. These naive CD8 T cells from the STAT3 mice really led to this very accelerated diabetes onset, much more so than wild type mice developed. And this was like, I think, the most striking finding.
That's almost a vertical line.
Yeah, I mean, it was amazing to us. So that was kind of the
Jeremy, how many mice was it? In the vertical, I forgot.
I want to say 10, maybe.
Yeah, it's pretty amazing. Yeah. All 10 mice get diabetes the same day after you do the transfer stuff.
Yeah. It was very powerful. Yeah. So to kind of summarize our findings, what we think is going on is basically STAT3 seems to lead to overall T cell activation and proliferation, kind of the shift from the naive to the effector state. More than that, whereas wild type mice, they might progress to the exhausted state, these STAT3 mice seem to get a little more locked into that effector and transitory state. And these cells, based on our single cell data and confirmed with our FACS data, seem to really secrete a lot of CCL5 chemokines like CCL5 and cytokines like granozymes, kind of your classical effector cytokines. And we think those are probably what's driving this more rapid destruction of the beta cells and ultimately accelerating autoimmune diabetes. So that's kind of the high level summary. And I know we've had some great discussions so far, but certainly additional questions, happy to discuss. Yeah.
I'm going to ask a question from the audience first, and then we are going to discuss things maybe. And now Otto asks whether complete STAT3 knockouts mice are still viable. Like, that's his question.
Like, are they STAT3 knockout, or like if we made ours, because ours were heterozygous, are you saying if we do homozygous STAT3 gain of function, are they viable? Yeah,
Like,
so that was
That's a question I'd like to know the answer to.
Yeah, we if I could count the number of times we tried crossing the STAT3 het to the STAT3 het to make these homozygous mice, it'd be a long list of times. I mean, what happened when we would do this is already these mice tend to get sick earlier, right? And so when you're trying to breed these mice, STAT3s with STAT3, they don't usually, they're not the most excited mice to take part in breeding. So that's hard enough to take place. And then on top of that, sometimes like during the pregnancy, the mom would develop diabetes because these mice had accelerated diabetes. And so that kind of screws it up. But sometimes they would end up dropping a litter. And I will say these mice, because they would often die very early, like within the first week or so. And so then I would have to basically genotype afterward what they were. And we did make some that were homozygous. They did survive and were able to be born, but they wouldn't survive very long. And so we didn't have an easy way to study them. Yeah, but I will say they were very small, and I suspect a lot of these findings would probably just be magnified significantly. But we had a really hard time generating those mice. But it's a really good question, because we, as Mark said, we've been wanting to know what that looks like. Yeah.
I have several questions, but I'll start with this particular one. This is a gain of function in STAT3 that's causing the disease. Do you think, like, would it be a possible experiment for you to check if a JAK inhib would work in this situation?
I think you've been talking to Mark. Yeah, that's another great experiment. I think Mark was maybe talking with Eli Lilly about obtaining one of these JAK inhibitors, because you would think that if you dampen some of that upstream signal. So first, it's important to point out this was a DNA binding mutation. It's a specific STAT3 mutation we inserted. So it is possible that even if you dampen that upstream signal, you still might see acceleration. But we do think it would probably be slightly less if you're not getting that initial activation of STAT3 via the JAK inhibitor. JAK inhibitor experiments, probably depends which one you use. They're also pretty painful experiments to execute, because I think some of them require like twice daily oral gavage of the JAK inhibitor for days and days. But even so, it's an interesting experiment that I think should get done at some point. Yeah.
Also, would you, like, I don't know if you are into mechanistic side, if you're interested in that, like, have you seen any changes in the CD8 positive cells with STAT3 phosphorylation status? Are they more or less activated?
Yeah, so I did phosphoflow pretty early on. I got kind of inconsistent results, and so I kind of didn't push that a lot more. Cytoph actually, it seemed to be the best way to look at some of these questions. But when I did phosphoflow using mass cytometry, I didn't see any populations that were lighting up and showing much higher degrees of STAT3 phosphorylation.
Was that tyrosine 705? The phospho?
Sorry.
Because so there's another very interesting site in STAT3 that is serine 727 site, and what that does is so I don't know if it's been shown with STAT3, but it's been shown with STAT1 that serine site, it helps in enhancing the interaction with the DNA binding domain. So I wonder if you have looked at it, if you have any hypothesis about it.
Yeah,
the interesting part is that serine site is not controlled by Janus kinases. It's actually controlled by certain MAP kinases, and even calcium associated kinases. So yeah, just something out
we,
there.
we have not done any of those molecular details at this point.
Yeah.
But
Okay.
one important point to make though is like, in vivo, when all hell breaks loose, right, and you start making this autoimmune response, right? Who's the cart? Who's the horse? Right? Huge problem. Right? That's part of the reason you have to make this mouse model, right? Because by the time the patients come to clinical attention, all hell is broken loose, and you don't know like what's the original thing that's caused everything to happen. So even in this highly controlled mouse model, it's really hard to tell what's going on because like Jeremy said, he did some STAT3 flow. It's not like there's like, oh, there's this one population of cells, right, that has hyperphosphorylation of STAT3, right? You find it in a whole bunch of cells. And it's probably just because, once the cytokines start getting made and things are firing, it auto amplifies and you get this sort of mess that's going on. Does that make sense?
Yeah.
Yeah. But your original question, a great one. I think that, targeted therapy to shut this thing down is a really, really good idea. So we're working on it. It's a heroic experiment, but I actually got drug from Eli Lilly to get this going finally.
Anyone else has anything?
Yeah, I had a small question. If we can, like, if this whole STAT3 cross can be done with a RIPO or a RIP-GP mice where we induce the diabetes, and do you think we can expect that like similar results in that model as well?
I don't know.
Yeah, I'm not sure.
I don't know. Why do people do that model?
Is that an inducible model? I've never heard about it.
Yeah. Well, I'll tell you why. Here's the dirty detail. Like, this is important, right? So, this is like the most contrived thing ever, right? You're like, oh, well, these B6 mice, they don't get diabetes. Okay, well, let's put an antigen in them, right? That islets normally never make, right? Oh, and then let's give them a Tcell at a high precursor frequency that can see that antigen, right? It's like a model of nothing, right? And the only reason that this is done is it's a way to make C57 black 6 mice get diabetes. And there's this whole army, in immunology, our workhorse model for knocking things out or monkeying with genes is in the C57 black 6 background and not the NOD background. So this is just a way for people to do experiments in B6 mice that have something to do with the word diabetes. Would be sort of my quick summary of it all. So
you're saying it's a horrible model to study diabetes.
Oh, horrible.
Okay.
Like what human being is walking around with, ova or LCMV all the time in their islets? It's like, it's so contrived. It's really bordering on worthless. But what would be an interesting experiment, that we have not done is you can make a C57 black 6 mouse that is black 6 everywhere in the genome, except at the MHC. And then at the MHC, it has the NOD's MHC, right? I would be curious what happens with diabetes in that mouse, because that mouse doesn't get diabetes either normally, right? But there you've got the right MHC and maybe, the right ability to generate diabetes. So I'd be very curious if that mouse got diabetes when you put the STAT3 thing in there. That would be cool. I bet they do. I don't know. What do you think, Jeremy?
Yeah, I think, if you're keeping that MHC in place, I think you're setting it up for following the same mechanism. So I would bet you're right.
Yeah, I actually think another cool thing about Jeremy's story, just to he did a beautiful job presenting all of this stuff to you, but that seems to be emerging with this diabetes, with this type 1 diabetes knowledge, both in humans and in mice, is in humans now. We've started doing these, for 20 years, more than 20 years, we've been doing clinical trials with different immune therapies to intervene and see if we can slow down type 1 diabetes, maybe even maybe even prevented in kids. And so one of the drugs that we use and use for that is this drug called anti-CD3 that tickles the T cell receptor. And one of the mysteries, of course, and it does slow down the progression of type 1 diabetes in kids that just got it, right? And so one of the mysteries has been, well, how does this drug that tickles the T cell receptor, what does it do exactly? And the data is emerging that one of the signatures you see in the blood of patients that get treated with this drug is their CD8 T cells go into sort of this exhausted state, right? And so it's very curious to both Jeremy and I that this model is sort of the opposite of that, right? The STAT3 seems to perturb the ability of CD8 cells to go into this exhausted state, and they just push through to the effector state. And that's probably why this accelerated diabetes stuff is happening.
So are you talking about OKT3, that anti-CD3 thing?
Well, it's non-mitogenic anti-CD3. OKT3, it's like OKT3, but then the FC portion of the antibody has been modulated, so it doesn't deplete T cells like OKT3 does. OKT3 was the original antibodies used by, it's still used by transplant surgeons for transplantation, and that depletes T cells big time, right? Whereas this non-mitogenic anti-CD3 is less depleting, but modulates the immune system, right?
Okay.
So where else is this, like, you guys are reading papers and doing journal club, where is this exhausted CD8 T cell stuff like exhausting all of us?
But you're saying why we aren't all exhausted?
Well, I'm exhausted because I can't pick up an issue of Nature Cell Science or Nature Immunology without some paper that involves cancer and has the word exhausted in it. I got it. How many versions of this story do I need? But I think it's kind of cool that, look, cancer, this cancer immunology and what's going on in type 1 diabetes, it's the same problem. It's just the switch, the switches are, we're trying to manipulate them in two different directions. But the key thing is the central tenets are probably generally the same.
Mm-hmm. There's a lot of advances in metabolism with respect to bringing back exhausted T cells, because in cancer they are bad, and in this case you want them to be exhausted so they stop all that destruction.
True. Yeah. So so this elegant switch that the immune system is using, right, to try to abort, basically try to abort a response, right, and how that plays out ultimately has a link to disease. It's pretty interesting.
Yeah.
Here I have a weird question. Like, is it possible to induce pancreatic cancer in a type 1 diabetic mouse model to actually see if the T cells are negating against one another?
Yeah, interesting question. I don't think that's been, like that kind of stuff, again, C57 black 6 mice, right? Just because you have the zoo of all the different knockouts and reporter mice and so on. So most people do P6 and not NOD, but our colleague at UCSF, Jeff Bluestone, he's been working on an NOD model of putting tumors in the NOD mouse to do that kind of stuff. But my recollection is the tumors they're not pancreatic tumors, right? Yeah, but I have wondered about that. Yeah, pancreatic cancer is a tough one, really tough.
So without taking any of your time, I'll just ask this last one. So this is going back to what Mark was saying about Tregs and the FOXP3 thing. So Mark, do you not believe X Tregs exist?
Oh, totally.
Okay.
Absolutely. But then if you look at those cells, right, they're not weakly making FOXP3 anymore. They just don't make it at all, right? And I think for the most part, it's a pretty stable population, and you don't want this stuff going haywire all the time and deprogramming and having T cells go into different lineages, particularly T regulatory cells. But yeah, it can happen.
Okay.
It can happen. I don't think it's very common though. Very common would be my response. I think that the opposite is, way more common, is you can make effector cells become, at least some effector cells become Tregs. Right. And that actually, to me, that makes a lot more sense. Right. You'd want that mechanism.
Yeah. So I think there are some viral infection models where they see X-Tregs a lot. I would want to see them in autoimmune diseases, whether they're there or not, but I don't see a lot of papers about them, at
Yeah.
least for now.
Yeah, the toughie with that whole, like conceptually, the box has been checked that, yeah, this can exist. The toughie with all of that is to say, oh, okay, the cell that I see that I think is an X-Treg, was it really a
Yeah.
Treg before? Was it really? Like, and how the hell do you know, right? I mean, it's like, right, you're inferring it from a fancy mouse labeling system, right? But it may be that they like flipped on FOXP3 a little bit. You know, that doesn't mean that in its past life it really was a full-blown Treg, right? And
Yeah.
you know, that whole field, you know, like you get into that argument, you know, you got to think about it for a minute. It's just like, oh my God, what a can of worms that can be. Because you know, if you play the doubting Thomas game, right, and you say, oh, I got these cells, and they used to make FOXP3, and you know, all this stuff happened to make them used to be FOXP3. The naysayers will say the following: it's like, okay, you know, when you purify these cells, you had contaminants, right? And there were a few contaminating cells that didn't really turn on FOXP3, and now you got this population that used to have made FOXP3, right? I mean, that's one thing. Or then if you do a transfer experiment with this stuff, and you transfer pure FOXP3 positive cells into a mouse, right? And then you find a few that converted, right? The naysayers will go, well, you had 2 cells in there that didn't have FOXP3, right? You just get into this contamination argument. There's no end, and there's no end to it. There's no end to it. This is the kind of nitpicky immunology I try to avoid.
Yeah. Okay.
It's not really helping that much.
So what are you guys working on next? Where is this study going to?
Well, I think that treatment with the JAK inhibitor is,
Okay.
I think, on the short list, right? That's super important. You know, you know, we had this patient case last year of not STAT3 gain of function disease, but another STAT gain of function disease called STAT1. And those patients also can get type 1 diabetes. And there's this, you know, this amazing case I was on at the New England Journal. These guys in Texas called me, or they had a patient who had got type 1 diabetes when he was like 15 or 16 years old. And he had a bunch of other clinical problems. And then a year after he was diagnosed with type 1 diabetes, somebody said, oh, something else is going on. And they sequenced his exome. And they figured out that this kid was actually a STAT1 gain of function patient, right? And because he's a STAT gain of function patient, they did put this patient on a JAK inhibitor, right? And so then what happened, right? And so these other features of the STAT1 gain of function disease include things like candidiasis and respiratory infections. And once he started on the JAK inhibitor, all of that stuff started getting better. And then he came to clinic and he told his doctor, you know, I don't think I need to take insulin anymore, right? And his type 1 diabetes went away. Oh. It actually reversed. And then that's why that single case is in the New England Journal, because that never happens, right? But that's super informative, right? And so then the big question, you know, that phenomenon is a big deal, but we have no idea why it reversed.
Yeah. So you're saying that the patient got cured?
Yes. He is now like, and now he's like, I think he's like 4 or 5 years out from his diagnosis, and he's not on insulin at all. And he had the autoantibodies, type 1 diabetes. He had DKA. He definitely had type 1 diabetes. There's no question.
Well, it's not
That's
often
pretty
we
interesting.
hear that word in autoimmune diseases.
Yeah. Well, in type 1 diabetes, you know, other autoimmune diseases, you can get the, you know, things tend to wax and wane, right? Rheumatoid arthritis would be a classic example. People, they'll have flares, right? Their joints will flare, then it'll get better. Joints will flare, it'll get better. Type 1 diabetes doesn't behave this way. You know, once people clinically come to clinical attention and they actually have diabetes, right? You know, it never goes away. And probably a big component of that is pancreatic beta cells are pretty wimpy at replacing themselves. They're very sensitive. They're very sensitive population of cells, right? Whereas, you know, the synovium in the joint, right, or in psoriasis, the skin, right? The skin can grow back, and repair itself much more robustly than the beta cells can. So it's an unfortunate part of type 1 diabetes is that the beta cells, I think it's a byproduct of the autoimmune process, right? It's just beta cells in general are not the hardiest cell to replace themselves.
Yeah.
Yeah, but that could, honestly, that could be a component too of what was going on in this kid. We don't know this for sure, right? I mean, I kind of poo-pooed that the beta cell effect of these stats is not the problem, and maybe that's part of what's going on. We don't know. Yep. So the mouse model is the key to figure out in vivo what's changing in the immune system when you give the JAK inhibitor, and maybe it would be kind of interesting to see in this Jeremy's model, does it reverse the diabetes?
Yeah, now you say that. So we recently did an experiment in NOD mice with a JAK inhibitor-like molecule that we have, but it was just on NOD background, and we were close to getting something working, but it eventually did not work. So these mice were not getting better. It looked like they were, but eventually they all died. So now I'm thinking this model that you have generated with a STAT3 gain of function, that might also work with what we are looking at.
Yeah. Yep. Yeah. And the cool thing is anytime you get the situation where you actually reverse diabetes, and then you can see what's going on, right? Once you get that phenomenon, then you can start teasing apart, well, okay, well, these are the things that are wrong, and these are the things that I'm reversing, right? And then you can start fine tuning it and probably move it into other systems like a full-on regular NOD mouse, or ultimately a patient that has type 1 diabetes. That's the way to think about it.
Yeah. Okay, so anybody else has anything else before we wrap up our session today?
I had just one final question just about, I'm just curious about the gamma delta T cells and this. Like, is there any case of anyone having any weird gamma delta T cells or infiltrating the islets or during type 1 diabetes, or they're just not there?
During our single cell RNA-seq, we did look at those gamma delta T cells. They were a very, very small population. No evidence that they were, like really any different from the wild type in terms of frequency.
Yeah, such an enigmatic cell population in general, right? And it's funny, the gamma deltas, they go through sort of waves up and down of like what they do and what they may not do. But in general, gamma delta cells are residents of epithelial tissues, and probably are involved in homeostasis in those places. But their life history and the type 1 diabetes story is not particularly convincing to me that they're a key player. Jeremy, you agree with that?
Yeah, no, I do. Because early on, I did a lot of facts as well. With seeing, you throw it in your panel and see like, is there anything interesting going on? And there's never anything very remarkable that I'd find.
Yeah, for you guys, I think this is a good lesson to learn, though, is these waves come along in immunology where a new cell population is found. Right. I guess you heard me hammering already about exhausted CD8 T cells in cancer, right? And so then, the problem is that like once that happens, people realize, oh, if I write a paper about this, it's going to show up in immunity, right? And the next thing you know, there's like 20 papers on the topic, right? And you think like this is like, the cause of cancer, right? And it's probably part of it. It's not everything. And what I'm getting at is there's like huge publication bias with like the waves that come around, and so before the pandemic and before this cancer immunology stuff, one of the things that, one of the cool things that came out of immunology were these lymphocytes called ILCs, innate lymphocytes, right? So they look like, they're basically the NK cell compartment is more complicated, right? There are other lymphocyte-like cells that don't have an adaptive receptor, T cell receptor, right? And you guys know this, right? There's ILC1s and ILC2s and ILC3s. Well, the next thing you know, this is the scourge of human biology, right? I mean, like ILCs cause all of these problems, right? I highly doubt it. It's just that they're an interesting cell that people have bumped into. Right. And so, this is one thing to keep your mind, keep your mind on, right? It's just these waves come, a cell gets discovered, and now it's going to be implicated in all these kinds of problems. Right. And maybe it does a little bit, but I highly doubt that this is like the switch that's causing all of the different problems in human disease, right? And then, then there'll be the next cell, and it'll be that, right? Tregs went through the same thing, really.
Yeah.
So it's just one thing to keep your mind on, because, when people write these fundamental immunology papers, right, always goes to a higher tier if you can show that this is relevant to something, right? So there'll be the last figure or a figure in there that, and I put it in some in vivo model like us, I guess, here with the NOD or EAE, right, or an immune response, right? But connecting all of that to like real human disease, I'm not so sure, right? For the life of me, I can't tell you, because I just brought it up. I'm not aware of an ILC human disease, right?
Yeah.
So it's a good lesson to look at the world. And then on the opposite end of the world, what's great about Jeremy's project is it started with a human being. Right? There are human beings walking around on Earth with this problem. Right?
Yeah, I think that's a valid point, that you want to have this translatability between the mouse model and humans, and it's hard to find that link. Like for you guys, it was quite lucky that the same mutation was able to generate a very convincing effect in NOD. Yep. Oftentimes, that's not the case.
Yep, that's right.
Okay, Mark and Jeremy, thanks a lot for giving us your Saturday morning. We're really glad to have you here.
So sorry it was late. The dog woke me up at 4 a.m. Like, slept through the alarm. I'm so sorry about that. But it was really fun, guys.
Yeah, thanks for having us.
Yeah.
Appreciate it.
Same for us. And with that, we will end our episode today. You guys will be able to...
Jeremy, can you stay on for 2 minutes? I just want to catch up with you. I haven't seen him in...
Oh, yeah, we can chat afterward.
Yeah.
I'll call you. Yeah.
Okay, sounds good.
Okay.
All right.
All right, guys. So you'll be able to watch this episode later on, and we will see you later in the next 2 weeks. Bye-bye.
Bye.
Bye-bye.